This video is to help my AP College Biology students do the assignment for describing protein structure. The assignment's listed in the Unit 1, Nature of Science and Chemistry of Life folder on Schoology. So if you'll go to your Schoology page, open that folder. You'll see the Describing Protein Structure assignment that tells you what you need to do, and then this it tells you to load this page, this PDF file, open it up, Describing Protein Structure PDF. So when you open the PDF file, here's what you see. You go online to the NCBI website, that's the National Center for Biotechnology Information or Biological Information. So we're going to click on that. It'll open up a new tab. Then you can toggle back and forth between your instructions and your tab at the NCBI. And it says from the drop-down menu in the upper left corner, select Structure. So that's this drop-down box right here. We're going to select Structure. And then we're going to search. It says in our instructions, in the search box at the top, type in the name of a protein. If you don't know a protein, Google to find the names of proteins. So you can do some internet search for that. So I'll open up a new tab and I'll search for, let's say, muscle protein names. Okay, a lot of you guys are athletes, so there's this one lists 4 different names for muscle proteins. So myosin, actin, troponin, tropomyosin. That's the scientific names for those muscle proteins. So let's go back to our NCBI website. I'll type in myosin in the search bar, and it returns these results. So these results show different research papers. It says here different scientific research articles about that protein. From the research article you select, click on the View in IC in 3D that is at the bottom left of the article. You gotta love whoever made that IC in 3D computer language. So this one, they give the scientific names of the species. This one was from a tarantula, so it must be some tarantula muscle protein. So that's fun. Let's click on this View in IC in 3D. See here? Each one of them has it in the lower left of the article, so I'll click on that. And when that loads, this shows this protein. This is a huge protein. You can kind of see it all coiled up with tons of little tiny alpha helix coils in there. This one's awful big. I'm thinking that's going to be too complicated to describe for right now. So you know what? This may happen to you where you pick one that's like, this is way too complicated. Just say, you know what, I'm going to go back, pick a different one. So let's go back, select Structure again, and pick a different protein. How about troponin? You know, the third one. So that's a different muscle protein. So I'll search for that. Comes back with, here's a human cardiac troponin muscle. So let's view that in IC in 3D. When it loads that, that's a much smaller. You can see some of the alpha helix. You see alpha helix coils. You can zoom out. You can zoom in. You can rotate it around to get a good view. Now the instructions here say, on the right side of the results page, click on Details to show the primary structure of the protein. So here's the right side of the page. I'm going to click on this Details, and it shows. Here's my protein 4Y99. That's the one I'm searching, so it just splits it up into, here's my different amino acids that it's built out of. It uses the 1-letter abbreviations for the amino acids, so D, D, I, Y, K. So if it says here to write down the names of the first 10 amino acids listed, I only need the first 10 just to teach you about the primary structure is the order of amino acids. But if I don't know what the letters represent, I'll have to look them up. So I can Google an amino acid chart that shows the names, or look them up in the textbook. So let's open a new tab. I'll Google amino acid abbreviations. And there's several different charts. I'll click on this one. It comes up, and here's the names, the 3-letter abbreviations, the 1-letter abbreviations. So when I look at my results, it was D, D, I, and then Y. So D. Find D in the chart, it's aspartate. So aspartate, then I was isoleucine, Y is tyrosine, and then the next ones would be KAA, so I go back, K is right there, it's lysine, A is alanine. Alanine, so you just write those down for the first 10 amino acids. That's the primary structure. Then the secondary structure, it says for number 5, for the secondary structure of the protein, count and list the number of alpha helices, beta sheets, and random coils. So I'd go in here and I'd say, well, I can see these 2 blue alpha helices and 2 brown ones here, or tan ones. Oh, and then a small brown one up at the top. So that's 5 so far. And now for the pink ones, down at the bottom I can see 1, 2, 3, 4, and then at the top 1, 2, 3, 4, 5. So I'm counting 9 of the pink alpha helices plus the 5, 3 brown ones, 2 blue ones. So that gives you your total number of alpha helices, 14 alpha helices, and there's no beta sheets in this particular protein. However, I can see a few random coils. The blue 2 alpha helices are connected to each other by a random coil, and same with the tan at the bottom, so that's 2 random coils. In fact, you can see there's random coils that connect each of the helices. So just count those up, and then 0 beta sheets in this one. Now for the tertiary protein structure, it says describe the overall 3-dimensional shape. And some of them have weird shapes. I don't know how to describe them, so maybe tubes or donuts or blobs or whatever. So to help you see the 3D shape, it says go to the top of the page, hover over Style and Protein, and select Sphere. So I go up to the top here, Style, I hover over that, hover over Proteins and select Sphere. And that's the shape. So it kind of looks like a long bent blob. Then for the quaternary protein structure, you can have the chains as different colors. It's already showing the chains as different colors. If it doesn't, you can hover over color and select chain, but as we hover over color, chain is already selected. And it shows 3 different colors, so that means there's 3 different polypeptide chains in that protein. Then you just need to write a paper with the name of the protein and the information from each level of the protein structure. Make sure your name's on the paper, and then either turn it in the tray or submit it on Schoology to me. Now let me do another example so you can see some beta sheets. So let's say we go back. Let's select, again, go to Structure at the NCBI website. This time let's do a different one. Instead of muscle proteins, let's do silk protein names. Okay. This tells me that silk is made up of 2 primary proteins. Got fibroin and sericin. So let's search for fibroin. Okay, if I search for fibroin protein, it comes up with different research papers about that. Let's click on the first one, View in ICN 3D, and there it is. Now those lines, those are beta sheets. We're going to count each line as a sheet. So, you can look at it like that and say, wow, there's 1, 2, 3, 4 of the kind of green sheets. There's 1, 2, 3, 4 of the blue, dark blue sheets, and then there's 4 of the pink sheets and 4 of the tan beta sheets. And then you can count also the random coils that are like this one on the edge here. So there's one random coil, another random coil here. So do your best. It may not be perfect, but do your best to say, hey, this is how many sheets and coils. And then again, for the tertiary structure, you go to Style, hover over Proteins, and select Sphere. And I would call that maybe a tapered blob. It looks like it's a little narrower on one end, so the top is narrower than the bottom. And again, if you click on Details on the right, you can see the amino acids in order using the 1-letter abbreviations. Okay, so hopefully that helps you in understanding protein structure much better.